Adrian, I think I could identify you. Jalen, I think I could identify you. Kiki, I could identify you. Castro, okay, so most of this class, but my other class, I do not know what they look like. So that's going to be interesting if and when we return to school.
I don't even know where half of these people's rooms are. Like, you know,
Right.
the school of the teachers.
Yeah, that's not surprising either. So we find ourselves in an interesting school situation where we are in October and don't really know what each other look like. But that's okay. You know, we've been doing pretty well. We've been adapting. You all are a resilient bunch, so I'm proud of you for that. So let's make sure we're starting our note-taking process by writing down the title of today's lesson. Today's date is October 27th, 2020, and it is Unit 7, Day 2. By the end of today, you will be able to predict ratios of complete inheritance by practicing problems. We're going to do a lot of practice problems today. And we want to answer the question, why do we use Punnett squares to predict offspring ratios? Has anybody ever heard of a Punnett square? You can just answer in the chat, yes or no.
Yeah.
Okay, so we've got 2 no's and a yes. So this will be a new concept for some people, but I'm going to lean on you, Gabby, to walk us through. No, I'm just kidding. All right. So we talked about the definition of inheritance yesterday. It is the process by which genetic information is passed on from one generation to the next. You inherit your genes from your parents. And the same is true not only for humans, but also for any other organism that reproduces. And so the purpose of Punnett squares is to simulate the process of this inheritance, to simulate random assortment and fertilization in order to determine potential ratios for offspring genotypes and phenotypes. I know you all are writing, but can someone just come off of mute really quickly and remind us what the difference between genotypes and phenotypes is?
Phenotypes is what you see, and genotypes is what you can't see.
Okay, I do like that way of thinking about it. So you can see
Can
a
I say
phenotype.
something as well?
Go ahead.
Was it pheno? How do you say it again?
Phenotype.
Yeah, phenotype. Isn't that the physical traits that you can see? The
Excellent.
Part of you, and then I don't know about genotype. I don't know how to explain it.
Okay. So I do like, it seems like we're kind of grasping onto this concept of phenotype quite well. Gabby said it's the stuff that you can see. Ayana says that it is the physical characteristics. This is true. So a phenotype we can describe with adjectives like fuzzy or prickly or darker colored. These are things that we can see with our naked eye. We can identify. Even things we can't necessarily see with our naked eye, like the characteristics of specific cells. Like this cell is more circular. This cell is more cubical. Those would still be considered physical representations, so they're phenotypes. The genotype, of course, we've got the word, the root gene in it. So a genotype is a description of the alleles, as we talked about yesterday. We represent alleles with letters. So in this case, we've got AA. Those are 2 different alleles right there, and this would be a genotype. Over here, we've got capital A, lowercase a. These are, again, 2 different alleles that represent a phenotype. You get 1 allele from each parent. What would this genotype be called? What would this combination of alleles be called? With 2 capital letters.
Dominant.
Good. And what's the word that comes before dominant? Oops, I'm sorry. You could just give me the prefix.
Homozygous.
Yes, homozygous dominant. So we've got the prefix homo, which means same. So in this case, we've got the same letter, a capital A, and a capital A represents the dominant allele, which means that this is homozygous dominant. What about this? These two lowercase a's, what would this be called?
Homo recessive.
Yes, excellent. This would be homozygous recessive. So again, they are the same, but they're both lowercase. And the lowercase letter represents the recessive allele. So this is homo for same, homozygous recessive. And then we've got two others which are mixed. We've got a capital letter and a lowercase letter. What would that be called?
Heterozygous.
Heterozygous. Thank you so much, Alicia. So hetero, of course, means different. We've got a capital letter, which is obviously different than a lowercase letter. So this is heterozygous. So we've got to keep this in mind. That's the language that we will be using as we move through this unit and as we continue on in the course. But these Punnett squares help you to determine the ratio of potential offspring genotypes and phenotypes. What we mean by this, each of your parents also has two alleles, but of course you only get one from each parent. So on the outside of my Punnett square, which is this kind of chart right here with 4 spaces, on the outside, I'm gonna write dad's phenotype. We're gonna write dad's alleles on the top and mom's alleles on the left. So both mom and dad in this case are heterozygous. They each have a dominant allele and a recessive allele. Mom has a dominant allele and mom has a recessive allele. Now what we're going to do is just watch them mix with each other. So if dad's dominant allele mixes or fertilizes mom's dominant allele, then we end up with an offspring that has 2 dominant alleles, and that offspring is going to be called homozygous dominant. If dad's dominant allele mixes with mom's recessive allele, then I've got an offspring that is heterozygous and therefore will have the dominant trait. If dad's recessive allele fertilizes mom's dominant allele, then again, I end up with a heterozygous offspring that expresses the dominant trait. And the only way that I get a child that has a recessive trait is if dad's recessive allele fertilizes mom's recessive allele, which gives me a homozygous recessive child. This is the only way that they can express a recessive trait. So in this case, my chances of having a dominant phenotype are 3 out of 4. I've got a 75% chance of having a dominant phenotype. I've got one dominant phenotype here, another here, and another here. So 3 out of my 4 spaces are the dominant phenotype. I've got a 3 out of 4 chance. But I still have this 1 out of 4 chance that the child develops a recessive trait. Of course, the process of fertilization is totally random. You don't know which sperm is going to meet which egg. You don't know unless you do some type of genetic counseling what the outcome of that fertilization would be. So this is why we use the Punnett squares, because the process is random, but we can figure out a ratio. We can figure out the probability of a specific outcome.
I have a question.
Yes.
So what is it called? Like, is it still? I mean, like a recessive gene is like a gene that doesn't. I mean, it can get passed on and stuff, but it's not necessarily like you don't have it, right?
If we are talking about complete dominance, and we'll kind of define these terms later on. But if we're talking about complete dominance, that just means that anytime the dominant allele is present, that person will express the dominant trait. So even look at this case right here on your screen, this GIF, this animation. Each of these squares ends up with one of the lowercase red t's. Do you see that?
Yeah, I see it.
But none of them end up with the trait of a lowercase red t, which I assume would be small. That's because the dominant the capital blue T is also present, and the capital blue T is going to dominate the lowercase red t. So even though I've got one of each, I'm only going to end up with the dominant trait. So that means that all of these offspring, there's a 100% chance that they are going to end up tall. They're going to express the dominant trait. But there's also a 100% chance that they will be carriers of the short trait, of the lowercase t. They carry this lowercase t in their genotype, but they don't express the recessive trait in their phenotype. So this is potentially getting to be a bit confusing. There's a lot of lingo being tossed out right now. So let's just continue on. I do want you all to write down these steps to complete your Punnett squares. It's always a good idea to start by making a key. So somewhere on the outside of your Punnett square, you would want to write capital T that equals tall trait, lowercase t that equals short trait. So make some kind of key to just to remind yourself what these letters and alleles represent. Your second step is to split the two parental genotypes on the outside of the box. So it doesn't necessarily matter where you write mom and dad. Mom can be on top, dad can be on the left, or vice versa, as long as you've got them split up. Then you're literally going to combine the alleles in the box. Combine the alleles in the box, in the boxes. And then you're going to answer the question. Whatever the question should ask of you, you need to be able to pick out that information from your Punnett square. So sometimes the question might be, what are the chances that the offspring of these two parents has the recessive trait? The question might be, what are the chances that an offspring of these two parents has the dominant trait? Another question might be, if four children, or if the chances of offspring having the dominant trait are 75%, then what must be the parent's genotype? So we'll talk about what some of these questions look like and how to answer them. But these are your steps. Thanks for joining us, Angel. I've got Omari, Ayana, Gabby, and Alicia here. I've got Adrian, Angel, Malachi, Stephanie, Kiki, Olivia, Jalen. Joseph, Castro, and Dante. Perfect attendance. Excellent job, people. All right. Does anyone need more time to write down these steps?
I don't think so.
Okay. So let's practice. We've got green leaves are dominant to yellow leaves. So our first step, as you all just wrote down, is to make a key. Make a key. So in this case, we choose the letter G. A capital G represents the dominant allele, and a lowercase g is going to represent the recessive allele. Of course, the dominant allele results in a green phenotype because we see that green leaves are dominant to yellow leaves. What's our second step? What is our second step? Did you all write down the steps?
Yeah.
So what comes next? Now that we've made our key, what should we do? Hello? Okay, let's go back. The second step is to split the two parental genotypes on the outside of the box. So I need to, do you guys need more time to write this down? This is important. No, I'm done. Okay, great. So maybe you all are just being a little shy today. But now we need to split the two parental genotypes on the outside of the box. So let's see what that looks like. Made our key. In this case, we're being told that the parental genotypes are as follows. Mom has well, you all tell me, what kind of genotype is this? Is it homozygous dominant? Is it heterozygous? What's that?
Homo-recessive.
It's homorecessive. Excellent. And then what about dad? What is his genotype?
Hetero.
Yep, dad's genotype is heterozygous. So we're going to combine them. We're going to write mom's on top. She's got homozygous recessive. So we're just going to write her two lowercase g's. And then we're going to split dad's genotype up as well. He's got heterozygous genotype. So one capital G, one lowercase g. Again, your capital G represents the dominant allele, which is green. So we highlight it in green. Now our Punnett square. Do we see how in a heterozygous genotype, we still express the dominant trait? So anytime I've got a capital G, it doesn't matter if there's also a lowercase g. I'm going to end up with the dominant genotype. The dominant phenotype, I'm sorry, it's going to be green. So just by looking at the Punnett square, before we even see what a potential question might be, just by looking at the Punnett square, what are the chances of an offspring between these two parents? What are the chances of an offspring being green?
50%.
50%, excellent. What are the chances of an offspring being heterozygous?
50.
50%, good. What are the chances of an offspring being homozygous recessive?
50.
Excellent. So in this case, it's pretty easy because the chances are always going to be 50%. So there's your genotypic ratio, there's your phenotypic ratio, 2 to 2, 50%, 1 over 2, or yeah, however you would like to say it. All right. We can also go in reverse when given the offspring genotypes to determine the parental genotypes.
Can you say it again? I didn't hear what you were saying. It was like
Yes.
cutting out.
So we can also go in reverse when we're given the offspring genotypes to determine the parental genotypes. So what this looks like is we have what's inside the Punnett square. We want to figure out what goes on the outside of the Punnett square.
A capital G?
Okay, so yeah, you're right. So let's break this down. So we should get in this box, we've got 2 capital G's, which means that we must have a capital G here, and we must have a capital G here. That's the only way we could end up with 2 capital G's here. Oh, it didn't go in the right order. And then in the other box, we've got 2 lowercase g's, which means that we have to have a lowercase g here and a lowercase g here. That's the only way that we could end up with a homozygous recessive genotype right here, is if these are both the recessive alleles. And the only way we can end up with a homozygous dominant genotype here is if these are both the dominant alleles. So what does that tell us about the parents? What do we now know about the parents' genotypes?
Both of them are heterozygous.
They're both heterozygous. And what does that mean about the parents' phenotypes?
They both have green leaves.
They both have green leaves, yes, because they're heterozygous, which means that they express the dominant trait. Excellent job. Okay. All right, so let's just go. So this box, we've got a homozygous dominant genotype. We take this A and drop it down. We take this A, push it over. We end up with 2 capital A's. What about in this box over here? What's my genotype going to be?
Isn't it going to be capital A and lowercase a?
It'll be capital A and lowercase a. Excellent. And what's that called?
Heterozygous.
Heterozygous, good. The next one is also homozygous dominant, and then the next one is also heterozygous. So what's our ratio here?
100%.
Okay, your phenotypic ratio would be 100%. We're looking for the genotypic ratio. So let's see what that looks like. We're gonna take these simple numbers. So we've got 2 of these, 2 homozygous dominants. We've got 2 heterozygous, and we've got 0 homozygous recessives. So that's what we mean by a ratio, just 2 to 2 to 0. Now, Alicia is right that phenotypically, all of the offspring are going to have the dominant trait. It'll be a 100% ratio for the dominant trait. All right, let's try this. So again, I'm just going to take the capital B, the dominant allele, and I'm going to shift it over. I'm going to drop this recessive allele down. So I end up with a heterozygous offspring. Same thing over here, heterozygous. Same thing down here, heterozygous. And same thing over here, heterozygous. And this time we're asking for the phenotypic ratio of the offspring. So how many, if brown is the dominant and blonde is the recessive, how many of the offspring are now going to have brown hair?
50.
All of them.
Okay. Can you explain, Alicia, why it's all of them?
Because the heterozygous ones always show the dominant trait.
Exactly. Anytime we have a heterozygous, and Castro, I appreciate you answering. But anytime we have a heterozygous genotype, as we do here, heterozygous meaning different, so capital B, lowercase b, it is going to express the dominant trait because this capital B dominates the lowercase b. So the brown hair dominates the blonde hair. So even though these are heterozygous, we still end up with all brown hair. Oh, why didn't it answer? Okay, oh well. So in this case, we would have 4 offspring with brown hair, 0 with blonde hair. So the phenotypic ratio is 1 or 100 to 0, or I guess you'd say 1 to nothing, 1 to 0, 4 to 0. 100% of the offspring are brown haired. Now, in this case, when figuring out the parental genotypes, you don't really need to look at these boxes in the top right or the top left. Just look at this box in the top left first, and it'll tell you what needs to go it will tell you what needs to go over here and over here. And then look at the box in the bottom right, and it'll tell you what needs to go over here and over here. So if I've got 2 dominant alleles, then obviously both of my parents have 1 dominant allele. Keeps doing that order like that. So what we've been talking, question?
Oh yeah. It was on the picture.
Okay.
How is that even possible? The tongue rolling?
There are mandibular muscles, meaning muscles that are like in your jaw that rest just under your tongue that allow some people to do it. Some of us don't have those muscles because we don't have the genes that code for those muscles. Just like some people don't have the genes that code for chin dimples or freckles or widow's peaks or attached earlobes.
Attached earlobes? Don't we
Yeah.
all have attached earlobes?
No, some people have hanging earlobes. We call those detached earlobes.
Oh, okay. Now I see what you're talking about.
Okay. So what we've been talking about so far is called complete inheritance. And what this means is that the dominant allele is always going to mask the recessive allele. It will always dominate the recessive allele. This means that You either have the trait or you don't. It also means that the heterozygous genotype is going to show the dominant trait. And this is what Alicia was just talking about. This applies when you have what's called complete inheritance. Okay, so here's the question. Brown eyes, capital B, are dominant to blue eyes, lowercase b. If a heterozygous brown-eyed man, meaning he's got one capital and one lowercase, is married to a blue-eyed woman, meaning two lowercase, what are the chances they will make a blue-eyed baby? So keep in mind that the only way to get a blue-eyed baby is by having a homozygous recessive genotype. That's the only way. Homozygous dominant gives us brown eyes. Heterozygous gives us brown eyes. This is complete inheritance. The only way to get a blue-eyed baby is with a homozygous recessive genotype. So step one, we make our key. The capital B represents the dominant allele, which is brown eyes. The lowercase b represents the recessive alleles, which is blue eyes. Our next step is to place the parental genotypes around our Punnett square. So we're going to put mom on the left and dad on top. Mom is BB. Mom is, homozygous recessive, lowercase b, lowercase b. Dad is heterozygous, capital B, lowercase b. So dad goes on top, capital B, lowercase b. Mom goes on the right, lowercase b. On the left. Lowercase b, lowercase b. Oh, God, this is horrible. And then we combine our alleles. And then we answer the question, what are the chances they will make a blue-eyed baby? How many of these offspring are blue-eyed?
Two?
Yes, two.
Two.
Yep, we got two blue-eyed babies. So what are the chances? Two out of how many?
2 out of 4. Oops.
Excellent. 2 out of 4, which gives us 50% chances. Good job. Let's try another one. Right-handedness is dominant to left-handedness. Cross, which means to do the Punnett square, a homozygous right-handed individual and a left-handed individual. What percentage of their offspring will write with their left hand? So our first step is to make a key. We're going to use the letter R. Capital R then represents the dominant allele, which is right-handedness. Lowercase r represents the the recessive allele, which is left-handedness. Now we need to find the genotypes of our parents. So if we've got a homozygous right-handed individual, what is the genotype? What is that person's genotype?
You said what is the genotype?
Yes, what's the genotype of this individual? A homozygous right-handed individual.
Uppercase R.
Okay, so it says homozygous, so we should have two of the same letter. They should either both be uppercase, they should either both be capitalized, or they should both be lowercase. But it does say that this person is right-handed, which means that that's the dominant trait. So that means that they both should be capitalized. And then the other is a left-handed individual, which means that they express both of the recessive alleles. So they should both have lowercase r's. All right, now we just assign those to places on the Punnett square. Typically, we do put, well, there is no father or mother here, but we're going to put the homozygous dominant on top and the homozygous recessive on the left. Then we do our crosses. So drop this R down, take this lowercase r over, we end up with heterozygous. Drop this R down, take this lowercase r over, we ended up with heterozygous again. Drop this capital R, move this lowercase r, heterozygous. Drop this capital R, move this lowercase r, heterozygous. So what are the chances, if we look at these four offspring, how many of them are left-handed?
0.
0. We don't have any left-handed offspring in this case. We've got nothing but heterozygous genotypes, which means that all of the children are going to be right-handed. How many more of these do we have? Okay, yellow is dominant to red in flowers. So we make our key. We're going to use the letter yellow. I'm sorry, the letter Y, not the letter yellow. I need to go back to elementary school. We're going to use the letter Y to represent the alleles here. A capital Y represents the dominant allele, which is yellow, and a lowercase y represents the recessive allele, which is red. We've got a red flower mating with a heterozygous yellow flower. The red flower, therefore, must be homozygous recessive. That's the only way we can have red, is if it is homozygous recessive. Homozygous dominant gives us yellow. Heterozygous gives us yellow. Homozygous recessive is the only way we get red, so we know that that has to be the parents, one of the parents' genotypes. The other parent we're told is heterozygous, so that's one capital, one lowercase. We place that into our Punnett square, and then we do our crossing. Heterozygous, heterozygous, homozygous recessive, homozygous recessive. So what's the phenotypic ratio? How many yellow flowers do I have? What percentage of yellow flowers do I have?
You have 2. Oops. Wait, no. Hold on. You have 4.
For what?
You have 4.
You were right the first time. We have 2 yellow flowers, and we've got 2 red flowers. The heterozygous genotype gives us the dominant trait, which is yellow. So yes, these are heterozygous, but we still get the dominant trait. These are homozygous recessive, so we end up with a recessive trait. So 2 red and 2 yellow. Oh my goodness. Let's play this Kahoot, and then I will leave you all. That'll leave you with about 15 minutes, I think. To wait, no, this ends at 11:25. No, it's 11:35. So yeah, that should leave you with 15 minutes or so to do the assignment for the day. Why is this not working? It's only 10 questions.
It's the sound of an explosive Mari. They're cousins.
They're having sex.
And the incredible drama gets going right now. Chris has been here before with Jay. You are not.
That's annoying. Sorry.
Sir,
Yes.
you said it's the wrong code.
Did you use the one that I? What'd you say?
I was gonna say if you copied and pasted it from the chat, you might have an extra space or something. I just typed it. But I don't know.
We've got to ensure Trump 2020 does not win.
Did you make that video? Or is that the video that Kahoot just randomly has?
Well, I did not make this Kahoot, but whoever made the Kahoot chose this video.
Oh, okay. Are we supposed to use our real names?
Probably, but I don't really mind.
Oh. My brother's saying something.
Okay, that's 11 people. I think we have 14 on the call. So if you haven't yet joined, you can join us in progress. The science of heredity of traits. I don't even know what. Oh, okay. The answer should be the word that is defined as the science of heredity of traits. Good. Genetics. Ooh, Jalen in the lead. Okay. Different form of a single gene. We should have 2 different forms of a single gene. That should be called an allele. Sorry, I think I skipped it earlier. I thought, yeah, that's on me. I apologize if you didn't get to answer. I think only one person didn't, though. Jalen, oh, Ayana jumps up. An organism with two different alleles for a particular trait. Two different alleles. Okay, so that should be called heterozygous. Hetero is a prefix meaning different. So we would end up with one capital letter and one lowercase. Good job. Observable characteristic or physical expression of an allele pair. Physical expression. Excellent. That's a phenotype, physical. Joseph in the lead. It's a good game. A segment of DNA that codes for a trait. What's that called? A segment of DNA. Good. Alicia makes her big move. Another name for a heterozygous organism. We didn't talk about this, but let's see if you all can pick up on it. Good. That one was tricky. I have not explicitly
I didn't
used,
even know that.
yes, I've never explicitly used the word hybrid, but a hybrid, of course, is something that is mixed. It has elements of multiple things. So in this case, heterozygous organisms have the dominant allele and the recessive allele. So we can refer to them as hybrids. Good job, Joseph. Let's keep going. States that random distribution of alleles occurs during meiosis. This is kind of a throwback way back to Unit 3. Independent assortment. Excellent. Good job. 7 out of the 10 of you got that correct. Excellent. Omari with the jump into the top 5. An organism's allele pair that codes for a trait, aka the letters. Good. So this would be the genotype. Ooh, steadiness at the top. Joseph is still on fire. The form of a trait that is masked, hidden by the dominant allele. Excellent. That would be the recessive allele. Okay, Olivia with a good jump. Last question. An organism with 2 of the same alleles for a particular trait. 2 of the same. Good job. That's called homozygous. Homo is a prefix meaning same. Let's see who our winners are. Olivia, good job. Jumped into third place. Alicia, second place. All 10 questions right. And number one for the first time, I think. Joseph, all 10 questions right. And you did it.
Good job, guys. You did a great job.
Excellent. So it seems like we're really understanding these concepts. I do think that these Punnett squares will be a bit tricky for you all, especially as today we only talked about complete dominance. So I do want to make sure that you all have ample time to practice. So take care of the exit ticket as well as the asynchronous assignment for the day. And I think you'll be in a good place going forward. We've got 13 minutes. I think that should be enough time to get them both done. Okay, folks, it is 10:35. I'm sorry, 11:35. You all are good to go. I will talk to you all tomorrow. I hope you have a good rest of your Tuesday.